Good evening, everyone. Welcome to online lecture series of Physiological Society of India. This is organized by Education Committee of Physiological Society of India. It is happening on every fourth Saturday of every month between 7 to 9 p.m. Each session, we are taking some specific topic of mostly of undergraduate interest, and we are trying to support the understanding, physiological understanding of some areas of interest, particularly of undergraduate students. This series is not to replace the regular classes taken by experienced teachers or of your student of your institute, but this is to give an overview so that your understanding can be better. It's a supplemental support for the regular classes of physiology. This is also targeted as a synchronous as well as asynchronous teaching, asynchronous learning. We expect that you will be benefited, students of physiology will be benefited, and we will get your feedback regarding your requirements, regarding your expectation from this channel, from this lecture series. Today, we are, as it is already announced, we are having two eminent teachers with us, Dr. Barnali Basu, Associate Professor of Department of Physiology, Surendranath College. She is passionate in teaching with expertise in biochemistry, molecular biology, genetics, system physiology, and has 18 years of background of supporting students, developing instructional plans and organizing and grading exams and tests, commended for sustaining effective learning environment through prepared classes and relevant assignments and consistently achieving classroom management and academic goals. Passionate in research on genetics of diseases, enthusiastic and eager to contribute in team success through hard work. Attention to detail and excellent organizing skills are her skills. Clear understanding of system physiology and training in genetic polymorphism and epigenetic study enriched her. She is motivated to learn, grow, and excel in academics. She has completed her MSc, PhD, and then she joined Surendranath College. She is known to many of you. I welcome Dr. Barnali.
Thank
Dr.
you,
Barnali, can you
Prashunda.
hear me? Yes.
Yes, I can hear you.
Yeah, good.
Am I
So,
audible?
yes, you are audible. And today your topic is basics of digestive system. That's what you have planned. So,
Yes.
without much delay, we should start now. You can share your slides with us.
Thank you, Professor Nayak, for this your kind introduction. And I like to convey my sincere gratitude to Physiological Society of India for giving this opportunity to say something about in front of students and others who are behind the screen. I have started to present. Is it visible?
Yes, your presentation is visible. You can do that hide part. Okay, you can go for this full screen mode.
Yes.
Yeah, it is visible, and you can check whether it is changing. Is there changes? Slide changes are working or not? You can check once. Yes, it's working. So wish all the audience and happy learning. And stage is yours, Dr. Barnali. In case there is any issue, I will come in and I will let you know. Otherwise, let it be smooth.
Okay. Thanks again, the Physiological Society of India, and thank you all the virtual audience in this festive session, festive mood in the Christmas Eve. You are here or listening to me. Thank you very much for that. I'm trying to say something, the basics of the digestive system, which are basically as per the requirement of the undergraduate student. We all know that the digestion is a process where we eat something and it digested through the different organ system and broken down into small pieces, then absorbed, and the undigested material excreted as the excretory product and feces. And the overall, the gastrointestinal system, we know, divided into two parts. One is the hollow tube portion, the alimentary canal, and another one is the solid structures, that associated glands, the salivary gland, liver, gallbladder, pancreas, like this. And we are all aware about this, that we mouth is for the chewing with the help of saliva, and some amount of carbohydrate are digested in the mouth. The esophagus, it not directly involved in the digestion, but is a very important organ which propel the food from the mouth to the stomach, the next stage of digestion, where to some extent the protein is digested with the help of hydrochloric acid and the pepsin, and then the food enter into the small intestine, then bile and pancreatic juice mixes with the food, then the main and the last organ system for digest completely digestion of food, and then food enter into the blood is the smallest form, and the pancreas and liver secreted the digestive juices. Is it not a simple process as we seen here? It is a complex process, but is very particularly each of the organ is a particular function for where by which our digestion, we can completely digest a kind of food. So these are the digestive functions. We all know that. The another important thing is that our digestive system is also help us in the immunity, because the gut microbiome present in the huge area, the alimentary canal is a huge area, a long area. The gut microbiome present in the digestive system is very closely related to our immune system, and both give the humoral and cellular immunity, because the only organ system, the digestive system, is the area where we meet the external thing, that is the food, and along with food the various microorganisms and other thing. So the killing these microorganisms is another part of this gut system. So this non-digestive function is also important. Now the every classify the whole talk into these two area, that is, one is the organ from the where the food is moved from the mouth to the esophagus to the stomach to the small intestine to the large intestine, and this movement of the food is along with the digestion is truly the biochemical process, help with the digestive juices, the saliva, bile, pancreatic juice, and intestinal juice, or the succus entericus. And all this process, that is the movement and the secretion of juices in the proper time, and to what extent and how much, is finally are regulated by the hormonal regulators, secreted from the different organs and glands, and also the neural regulators, very closely related, a typical and very interesting neural system present there in the digestive tract. This is the overall picture of the digestive system. And let's go for the another important function before entering particularly the digestive system, that is the wall of the digestive tract. It's a peculiar type of wall, and the features are common from the very beginning to the end, and the four layers are present from inside to the outwards. The first layer is the mucus layer, and in the mucus layer there are the first part is the epithelium. The epithelium is the direct contact with the food. So this epithelium in the different part, the esophagus, stomach, the different segment of the small intestine, we will see that drastically the epithelial part drastically changes its character and secrete different things of materials for the proper digestion of food. And after that epithelium, there is a basement membrane on which the epithelium sit, and this epithelium is again sit on a connective tissue layer called the lamina propria. This connective tissue layer is an important layer. It has some blood vessels and some connective tissues which have the important functions. And after that lamina propria, there is a thin layer of mucosa, that is this is called the muscularis mucosa, because smooth muscles are present here, and the smooth muscles which present in this muscularis mucosa is very important in case of villi, because these mucosa smooth muscle are responsible for movement of the villi in the intestine. And after this thin layer there is a thick layer which is called the submucosa. This submucosal layer is also has different types of cells according to the need of the that organ system. And after this mucous layer, the outward layer is the come the muscle layer. And the muscle is very interesting. One is the present in the circular manner, the circular muscle, and another one is the longitudinal muscle layer. When the circular muscle contract, the this tube, the alimentary canal, shorten its diameter, but its length remains same. Whereas when this longitudinal layer contract, the diameter remains same, but the tube shorten. So by this way, the movement of the food is going on. In the later half of the lecture, Dr. Maitra will elaborate you about the motion, the movement of the food. So I am concentrating on the mainly on the digestion portion. These are the layer which I already told, and the outermost layer is the serous layer. This helps in the attachment of the surrounding structures with the GI tract. And our first digestion start with the mouth. After taking a food, with the help of tongue, we chewed with that tongue, we sense the taste, because the taste buds present in the different part of the tongue. With the teeth, we have different types of teeth, the incisor, which are we can cut, with the canines through which by which we can tear, and the premolar which we grind like a mixer grinder actions. And along with that, another important thing is saliva, secreted from the salivary glands, which helps the food to lubricate inside the mouth. And the salivary glands are the first digestive gland, and these are exocrine in nature. We have three pairs of digestive gland, salivary gland. One is the parotid, present just beneath the ear, and some mandibular in the under the jaw, and the lingual under the tongue. And recently in 2020, a fourth pair of gland was reported by a group of cancer scientists of the Netherlands, and they proved that another pair of gland present in the nasal cavity, named it as a tubercle glands. They proved with for a cancer patients when they cancer patients with the radiotherapy. They did in 2020. This is a picture, a detailed picture of the salivary gland. Salivary gland has two type of cells, acinar cells, that is the secretory cells. One is the serous acinar cell, and the main function of which is responsible for breaking down the food, and the mucous acinar cell which secretes mucin for the lubrication. And the salivary, each of the acinar cells are connected with the duct, and the ducts, all the small ducts are connected with a big duct, and saliva flow through the duct and enter into the mouth. And but the parotid, submandible, sublingual, each gland has a different types of, quite different from each other. In case of parotid, most of the cell are the serous type, and the main thing to secrete is the alpha amylase enzyme present in the tyalin, which is responsible for breaking down the starch to some extent. And so it is, the mucus is less in this fluid, so not so much fluid is present in this. And the infection, mumps, it's a viral infection, is caused in this parotid gland. And the next one, the submandibular. So what is the difference with the submandibular gland with the parotid is that this is a true mixed gland, the seromucous gland. 50, almost 50-50 composition, the serous and mucus. So they both secrete the serous, that is the enzymes, and also the mucin. And the sublingual gland, which is chiefly the mucus, though sometimes 25%, about 25% of the serous acini also present in the sublingual gland, but the main function is to secrete mucus into the mouth. So the composition is obviously the mucus, and very interestingly, WBC epithelial cells and some antimicrobial agents. Among these antimicrobial agents, very important is the lysozyme and the lactoperoxidase, lactoferrin, and also they secrete the immunoglobulin, IgA. They, because the food is enter from the external environment, and they first encounter with our body system, so we need to protect ourselves. So our body system is designed in such a way that is a number of players are there to kill the microbes enter into the mouth. Now the regulation, we observe that saliva can secrete even in absence of food. We know the Pavlov experiment of scientist Pavlov. The ring of the bell can secrete the itself from the stomach. Like this, if we see the food, sometimes that stimulus, the smell, obviously the taste, when enter into the higher centers of the brain, from the higher centers, the signal goes to the salivary gland and secretes saliva. So there is a very coordinated manner of the regulation of this salivary secretion by both the parasympathetic and sympathetic nervous system. Then come to the pharynx. Pharynx is the junction of mouth to the esophagus. And as it is a junction of mouth to esophagus, pharynx has a three part. One is the nasal part connected with the nasal cavity. Another one is the oral part or oropharynx connected with the mouth. And another one is the laryngeal part or larynx, and which is the larynx, after which they are connected with the respiratory tract. And after the oropharynx comes the esophagus. Esophagus is a long fibromuscular tube around 25 centimeter long. And the esophagus, the important and interesting feature is that the muscle layer of the esophagus is a transitional state from striated to smooth muscle, because esophagus need a strong muscle to contract and to propel the food and by the peristaltic movement from the mouth to the stomach. And near the stomach, it has a sphincter, where it connected with the stomach. Then when food enter into the stomach, this is the structure of the stomach. It can be, it is divided. One is the cardiac portion near the heart. This is the bulging portion is called the fundus. The main portion, the major portion is the body, and the lower portion is antrum. The different portion of the stomach is quite different from each other in their secretory nature and the function. Though the finely tuned, all the secretions in the stomach is regulated by one another. This is a histological structure of the section of the stomach. The epithelial cell layer in the first layer of the digestive tract in the stomach thrown into fold. It is called the rugae formation. And this rugae formation is quite different from the cardiac region to the fundic region to the pyloric region. And there are the cells present, the gastric glands, which these gastric glands secrete the hydrochloric acid and also the pepsin, which is required for the protein digestion. So in the gastric cardiac area, the most of the cells are mainly secrete mucus. Whereas in the intermediate zone or the body area, the parietal cell present or the oxygenatic cells, which secrete hydrochloric acid. Another one is the chief cell, the zymogenic cell, which secrete the enzyme pepsin and renin. Pepsin is responsible for digestion of protein with the help of HCl, and renin is responsible for curdling of milk. And also some amount of mucus also secreted from the neck area of this intermediate cell. And the pyloric portion, that is the end portion of the stomach, secrete mainly the gastrin, a hormone, a small digestive hormone secreted from the G cell. This is a elaborated structure of a gastric gland. This is called the gastric pit, the inner portion where all secretion gathered. This is the mucous neck cell, which secrete mucus. This is the chip cell, which secrete the pepsin and renin. And the parietal cell is the site for synthesis of hydrochloric acids. And each of the cell have different histology, and particularly as a different for the particular function. And the gastric juice mainly composed of many electrolytes, mainly the hydrochloric acid, the enzyme I've already told, pepsin, gastric gelatinase, some sort of lipase and amylase are also present there. And this is in the the parietal cells, the hydrochloric acid produced in a manner that against the concentration gradient, it can synthesize and accept the hydrogen ion, and it propel or eject out into the lumen. To prevent themselves, the stomach lined by a layer where the mucus is present. This is the function of the mucus gland of the stomach, to secrete mucus and prevent his own home, because the hydrochloric acid is secreted from the parietal cell, and this hydrochloric acid can digest the layer of this epithelial cell. However, the hydrochloric acid also secreted in a manner of jet manner expel into the lumen. And as this is a ATP driven process, so it can happen against the concentration gradient. The hydrochloric acid is synthesized, and bicarbonate ion extruded out from the cell. And in this process, the continuously hydrochloric acid is going on to synthesize. And this synthesis of this hydrochloric acid is finely tuned by the other the neurotransmitter acetylcholine, histamine, even in the gastrin. Now, the interesting feature is that same rugae looks like different in different part of the stomach. This is the cardiac portion, where the main cell is the mucus. This is the body or fundus portion. The main is the parietal cell, chip cell, and mucus cell present there. This is the main portions of the stomach, where the food is churned and digested, the protein is digested. And the antrum portion, the main function, the lower half of the stomach, to secrete the gastrin and some amount of mucus. Gastrin is a hormone which is essential for digestion of food in the stomach. Now, how this regulation is going on? The parietal cell causes the acid secretion, The acetylcholine, histamine, gastrin, the three players for which are actually which are regulated the secretion of hydrochloric acids and the other pepsin. Acetylcholine is secreted by the vagus nerve. Vagus nerve present outside the stomach, lined about the innervated into the stomach, and the stretching of the layer by the food can stimulate the vagus, and now acetylcholine is secreted. There are acetylcholine receptors on the chip cells and the parietal cells. They can take the acetylcholine and it causes the stimulate the hydrochloric acid secretion, also the pepsin secretion. Another is the histamine. Histamine is produced within the stomach by the enterochromaffin cell, ECL, in the body region, and is also acts as a paracrine fashion, that is secreted, and there it acts on its own cell. It also stimulates the hydrochloric acid secretion, but the gastrin is secreted from the lower half of the stomach, and this the gastrin to some extent causes regulation. There are three phases of digestion. One is the cephalic phase in the stomach, the gastric phase, and the intestinal phase. Actually, the secretion of the hydrochloric acid start even without food. Again, I like to say about the experiment of Pavlov. The ring of the bell can stimulate the hydrochloric acid secretion. That is the sensation of food or looking into the food. Without any food, it start to secrete the hydrochloric acid. It proves that the digestive system is closely connected with the central nervous system, and parasympathetic and sympathetic innervation is responsible for the and essential for the proper digestion. After entering the food also again and added another stimulus, that is stretching the wall of inside the wall of the stomach, and the histamine and gastrin start to release and start to stimulate the hydrochloric acid. At the end stage, that is the intestinal phase, when food is almost digested into the stomach and near to complete, and trying to enter into the intestine, that is the duodenum portion. Here, from the duodenum, cholecystokinin, secretin, gastrointestinal peptide, these are the small peptide molecules, small hormone molecules, they come to play. What they do? They do that inhibit the secretion of hydrochloric acids and other secretion. And because there is no need of the further secretion of this, and the food is now going to enter into the intestine. So all these three stages are finely tuned in the stomach. Another important cell present in the antrum portion, that is in the last portion of the stomach, is the D cell, which release the somatostatin. And very interesting, D cell is a inhibitory kind of activity rather than the G cell. Both G and D present in the antrum portion, but G stimulate the hydrochloric acid and pepsin secretion, whereas D inhibit this secretion. Both are act to the GTPase system, G receptor system, but the gastrin act as a stimulatory molecules, and this somatostatin acts as a inhibitory molecules. After the stomach, when food trying to enter into the duodenum, here the another digestive gland, the pancreas, comes to play. The pancreas secrete the two types of things. One is the exocrine secretion, that is enzymes required for the digestion. Another is, we know that, the endocrine secretion, that is the insulin, glucagon, somatomedin, like this, the hormones, somatostatin. This is the elaborate structure, that is the endocrine part or the acinal cells, from where the pancreatic juice secretes, and the pancreatic juice contains all types of enzymes which is required for the complete digestion of carbohydrate, complete digestion of fat, complete digestion of protein. And this is the basic composition, where all enzymes are present, the proteolytic enzyme, lipolytic enzyme, amylolytic enzymes, and also other the inorganic substances. And important inorganic substance is the bicarbonate, which is very essential. Why? Because to neutralize the acidic nature of the food which comes, the chyme come into the duodenum from the stomach. So the after entering the duodenum, the pancreatic enzymes mainly activate the uh, enzymes, that is trypsinogen to trypsin. All the enzymes are activated into the duodenum part and cause the complete the digestion. Now the regulation of pancreas is depends on the activity of the intestine also. When the food enter into the intestine, the duodenum, from there, I have already told the secretin is a cholecystokinin absorbed, secreted from the intestine and absorbed into the blood. They enter into the vagal stimulation, the area of the higher center of the brain when cause the vagal stimulation. This vagal stimulation causes stimulate again the pancreas. Then it secrete more amount of pancreatic juice. So there is again the inhibited by the secretin. So a very coordinated manner, the regulation of this pancreatic juice is going on. Then another structure in the left side of the duodenum, in the upper half the stomach, in the right half the pancreas, in the left half the liver. Liver is a very important organ of our body, and we know that it has a right lobe and left lobe. The main function of the liver is the synthesis and secretion of bile. Bile is not only help the metabolism of fat, but to some extent it has some other functions also. We'll see that it also helps the in the immunity process, in the cell signaling, and now the researchers reported that is a bile is a very important component for the nuclear receptor stimulator in the family of nuclear receptors. The main composition of bile is the bile acids. About 61% of this is the bile acids. And these are the salts of bile acids, which when added to the bile, the conjugated bile acids, the taurocholic acid, glycolic acid, taurodeoxycholic acids, there are different form of the bile acids. They are very important because bile acts as not only as an emulsifier, of that is the emulsification of fat, because when the food enter from stomach to the duodenum, till now the fat is remain as it is, and when the fat encounter with the bile has the two component. One is the hydrophilic area, another one is a hydrophobic area. The hydrophobic area attached with the fat globule, and the hydrophilic area attached outside with the water soluble part of the duodenum. So this forms the globule, and this in this way, the between the bile salt, the small enzymes can enter into the fat droplets and can broken down into the small molecules called the micelles formation. And when the molecules are small, these can enter into the broken down into the monoglycerides, fatty acid, with the small fatty acids, other without forming this, the fatty acid cannot pass through the intestinal membrane into the blood, and just trans crossing this membrane, this area, again these fatty acids, the small fatty acids, again they bind with each other and form the triglycerides and others. But to pass from the intestine to the blood, that is inside the blood, it is very essential to broken down the fatty acid in the smallest molecule, which is not possible without the bile, specifically with the bile salts. Now the liver is the circulation of livers is also finely tuned. These are the hepatic cells, the hexagonal cells, and the bile secreted from the synthesized and secreted from the cells. These are the bile ducts, and there is a central vein. And when they combine together and form a major bile duct, and this bile then enter into the gallbladder. Gallbladder is the storage area of this bile, and then enter into the duodenum. There is a special type of circulatory system that is called the enterohepatic circulation. Entero means the circulation going through inside that particular area. This is very important because it prevents the loss of bile through the excretion. So the bile secretion is after entering the duodenum, or some portion of the bile is present in the duodenum, and a major portion and again absorbed, and the rest of the portion, which is a few portions which is absorbed into the blood. When that blood enter passing through the kidney, so in the renal artery, in that area, bile again absorbed inside the cell. So bile is absorbed from this area, this bile again transported to the liver. So they prevent the loss of bile, and the loss of bile, because the bile is not only the essential for fat digestion, it is also essential for absorption of the fat dissolving vitamins A, D, E, K. So this is one of the reason when in the region where the child are malnourished or undernourished and suffering from the diarrhea, there is also the deficiency of fat soluble vitamin. This is a very important issue in case of child malnutrition. So after this the come to small intestine, food enter into the small intestine. We know that small intestine is a long tube, 25-meter long tube. Sorry, 25, the intestine, middle part and the all part all together. And the primary, the area is called the duodenum, the middle portion is jejunum, and the later portion is ileum. The interesting fact is that the villi is a three-tier system for increasing the surface area. The main function of the small intestine, the complete digestion, and after the complete digestion, the complete absorption of the digested food. So for the digestion, we know that the surface area is a big issue for the osmosis process. And so there is a three-tier expansion system of the villi. One is the microvilli present in the enterocyte, that is the epithelial cell which is in direct contact with the lumen. These are the microvilli. And after that microvilli, in the lamina propria, there is a villus, there is an area. Again, this is the folding which increases the surface area. And after the villus, there is the area, the submucosa, which is called the plici circularis. Plici circularis is a very important area where the villus is again thrown into folds in the submucous layer. So there is a three-tier system for increasing the surface area in the villus portion. Now the interesting fact is that this villi, the present glands present in the villi also differ, like in the stomach, from region to region. The basic glands are the goblet cells which produce the mucus, secrete mucus, they protect the epithelial layer, and the another one is enterocytes. These enterocytes are the cell from where the secretion occur for the digestion, and also they absorb the food and nutrients and ions. And the another important cell present in the area of the crypt of Leverkusen. Crypt of Leverkusen is the area, actually the basement area of the in the villus region, this basement area. And this crypt area, there are some important cell present. Uh, in spite of the digestion, other than digestion, another important function of this area is the the stem cell, production of stem cell. That is the continuous production of enterocytes, goblet cells, epithelial layers require the stem cells. And this crypt area is the area of stem cells which continuously supply the cell to the basal lamina. And two interesting cell also present there, that is called the enteroendocrine cell in the crypt area. One is the eye cell. Eye cell is responsible for secretion of cholecystokinin. We found that cholecystokinin is a very important small hormone which regulate the pancreatic secretion. Another is the S cell, which secrete the hormone secretin, and which is also responsible for secretion of the pancreatic hormones. So crypt, goblet cell, and the enterocytes present throughout the intestine. But there are the different types of secretion, the nature of the secretion and different from the duodenum to jejunum to ileum. The pH is slightly basic because after entering into the intestine, the food from stomach, it mixed with the bile. So this pH is slightly alkaline in this area. This is the area of duodenum. And the primary cell of the duodenum is the goblet cell, crypt, the cells in the crypt. And the important feature is the presence of Brunner's gland. This Brunner gland present in the submucous area and helps the secretion of mucus and other secretions. And after that duodenum, the jejunum. The jejunum is the area where the highest amount of absorption takes place in the intestine. And the maximum number of villi present in the millions of villi present in this jejunum. And the villi is highly tortoise form, that is increases surface area. There is in the submucous layer, there is no Brunner's gland. And in the ileum, there is another cell, the Peyer's patch. There is a no Peyer's patch. So this is the identifying features of this jejunum. Interesting is that in a negative point, that is the absence of Brunner's gland and absence of Peyer's patch is the characteristic feature or identifying feature of the ileum. However, the Brunner's gland is the identifying feature of the duodenum, and the ileum, the Peyer's patch, these are the lymphoid cell present. And the villi are, now the villi are quite the broken villi, and the sorry, there is spelling mistake, this is the Peyer's patch. Broken villi, the absorption is less than the jejunum, but the main absorption is the ions and water absorbed in this too with the food. And this certain, definitely there is the regulation of the secretion, the local stimuli obviously present, the food, the chyle is mixing inside the intestine. So this causes the stimulation, stretching in the wall. And there is a peptide, small peptide called the vasoactive intestinal peptide, that secretes the crypts of in the area of the crypts libercules, and it also causes the secretions of these enterocytes. And the Brunner's gland, also the Brunner secretion of Brunner's gland causes the stimulation of vagus nerve, which again important for the stimulation of the pancreas and to some extent the small intestine also. Then after the complete digestion, the some amount of food which we could not digest is come into the large intestine. But along with food, some important materials, huge amount of water molecules, some ions, these and glucose also come into the large intestine. So large intestine is also area of absorption of the water. And this is a ascending portion. The first portion which move forward is the ascending portion. This is the transverse portion and lies horizontally, and this is the descending portion. These are the three portions of the large intestine, and the end portion is called the rectum, which connected with the anus, from where you can excrete the feces. Now the large intestine, the wall of the large intestine is also a peculiar feature, not like the villi, but thrown into folds, and the maximum glands present there is the goblet cells and the cells for the water absorption. Because goblet cells are very important here, because the water is continuously absorbed in the large intestine. So we are continuously dehydrating the waste product. And so to prevent the wall of the large intestine from the mechanical friction of these dehydrated waste products, it required the mucus to lubricate the wall, so that wall cannot be damaged or due to the frictional force. And it, along with the water, it also absorbed the small ions. I've already said the potassium ions and some sort of vitamins are also absorbed in the uh, large intestine. Now, the important feature is that the digestion is only possible when there is a fine tuning between the central nervous system and the wound nervous system of the digestive area. That is, the wound nervous system is called the intrinsic component or the intestinal nervous system. This ENS is sometimes called the second brain. This second brain is present in the intestine. And this second brain is a two component. One is present in the outside, in the submucous layer. Between these muscle layers, two muscle layers are there. Between these two muscle layers, there is a section of the nerve that is called the myenteric plexus. The plexus, that is the ganglionic cell of this nerve, which originate in this area. Another one is the above the submucous layer, that is the Orbach Meissner's plexus. And this, myenteric plexus is called the Orbach plexus, and another one is mesenteric plexus. These two plexus actually causes locally control the regulation of this movement, the propel of this movement. However, from the central nervous system, the vagus and other nerves, the parasympathetic and sympathetic nerve is finely tuned with this system. And an important cell in this area is the cell of Kajal. The according to the name of the scientist Kajal, this is the called the pacemaker of the intestine. These cells are thought to originate is the impulse in this plexus system. So this coordinated system, the two system of the interstitial cell system, the ENS, the myenteric and Meissner's plexus, and the sympathetic and parasympathetic system, as I have told earlier, the vagus nerves, and this coordinate in a way that the food is completely digested and smoothly we can absorb it properly in a proper manner. So another important thing, that is, I have already told, the important function of the digestive system, other than digestion, is to maintain the microbiome, that is the gut microbiota, because gut is closely associated with the brain. So gut inflammation or gut microbiota maintenance, it not only depend on the immunity, it also depend on the nature of the food, the way we digest, how efficient our digestive system is. Depending all this, how can we absorb, then how efficiently we digest a food, how much a food stay inside intestine in which area, this all depend on the health of the gut microbiome. And this gut inflammatory signals from the gut microbiome is as a close connection with the brain. And in the many disease, it can say that in many neurodegenerative disease, there is a symptoms in the gut area. And in many gut area disease, the symptoms present in the central nervous system. For example, irritable bowel syndrome, IBS, is this kind of disease. And in some neurodegenerative disease like the Alzheimer's or Parkinson's also show some clinical symptoms in the this area, like the constipations and the problem in the defecation like this. So it proves that there is a close connection of the digestive system and specifically the gut area with the brain area. So the digestive system, as we see is not only for the just for the digestion and absorption of the food. Digestion absorption is related with each area of our body. So a healthy gut can not only give you the energy, it can give the it's essential for healthy immunity, and the immunity is essential for healthy brain and healthy mind also. Without a healthy body, a healthy mind cannot grow. So maintain a healthy gut, eat healthy, and digest in a proper manner by maintaining a proper lifestyle, so we can be stay healthy. Thank you all for the patience hearing for me. Thank you again, the Physiological Society of India, for this opportunity. Thank you very much. Over to you, Professor Nayak.
Thank you, Dr. Barnali. It's a great one, and you have covered so many areas, almost every part of the digestive system you have touched upon, and you have rightly said about the healthy gut, staying healthy. Thank you very much. And for the students, I would like to mention that this is just an overview of what about the digestive system. This is not a replacement of your regular classes. This is just an overview of the basic system of the digestive system. In addition to these basic information, you need to go in detail about the each and every part in detail for this system. And for that, if you have any queries, you can directly contact. I think, Dr. Barnali, have you shared your email ID, or would you like to share? Or other way also is there, like we can divert
Yes, I
if
will
we
share
get.
in the chat box, YouTube chat box. They can contact
Oh,
and they can put any question in the chat box, or if they want to know the each slide, this presentation, I can share it also,
okay, that's great.
because
So,
each of the
e-
slide has a link from where they can know in future they can know more in the slide.
okay, that's great. So,
I can
audience,
share it. I'll share the
they,
mail if anybody interested. I can share it, the presentation.
here is a compliment for you. Rajat Mukherjee has conveyed thank you for your nice presentation. And Dr. Sival Srivastav has also mentioned about
Thank you, sir.
PSI team for this informative lecture. So there are some feedback,
Thank
good
you very
feedback.
much,
It's
sir.
nice. It's encouraging for us.
Thank you all.
They can, if any query is there, they can directly contact you, as well as they can send information to us, and we will
Sure.
revert back to you. Thank you, Dr. Bernali. And now
Sure.
we
Thank
will
you very
be,
much. Thank you.
Dr. Rajan Haldar also mentioned it's a nice presentation. Ronit Tusir also mentioned thank you. So good feedback we are receiving. Lavanya, just a minute. Dr. Arjun, you are online
Yes, yes, sir.
and you can uh, uh, you can share your slides. Dr. Arjun Maitra, once a university gold medalist at graduation, did his PhD in medical physiology, and his area of interest remains in the domain of pulse and wave analysis and arterial stiffness. He has been in academia for almost 20 years. He has a keen interest in experimental physiology and neurophysiology, developed neurophysiology labs in different medical colleges, and led the team. During the COVID era, he successfully developed molecular testing facility and worked as a clinical scientist and received acknowledgement for the same. He has several publications and one international book chapter in his quiver and was selected as a reviewer for the BMJ group. As from the introduction, we know that he is a keen, interested teacher. So I wish we will be having a great session now. Dr. Arjun, your slides, I'm posting them. Yes, it is visible now, Dr. Arjun.
The slides, yeah, I think the
Yeah,
slides are visible now.
slides are visible,
Yeah,
and you can check whether they are moving forward, backward if you want. You can check once. Yes, it's changing.
okay.
So, Dr. Arjun, stage is yours.
Thank you, sir. I'm really thankful to the whole PSI team for giving me the opportunity to take this lecture on GI motility. So, let's start with that. What is GI motility? So, as we go on, when we talk about GI motility, the first thing is required that we should know that all the hollow visceral organ uh, is made up of muscles, starting from, except you can say it is the lung, which is not made up of muscles primarily, rather than normal tissue. But every hollow organ has got its own movement because it has to either receive, or it has to either move its content to and fro, in and out, and etc. Now, just like the other organs, our GI system also has a motility. For what? For movement of food. But now the question is, why do we require the movement of food? Because if we see the GI tract throughout, then you will find that it's a very huge, long network, and this tube, it folds for several times in different regions, and because of these foldings, sometimes the food moves towards the gravity, some movements are anti-gravity, and some movements are parallel to the gravity. So when it is anti-gravity, or it is the movement of parallel gravity, then it becomes impossible for the movement, because unless and until you need, or you provide some force, or to push the food material through the tract. We should also remember that these structures are made up of muscles and other living tissue, and they have its own elasticity. So they are not rigid tubes, but they are elastic tubes. So if there is no intrinsic mechanism to push the food forward, then they may they may increase, and they may hold the food for longer time inside the and the and the tube may get clogged. So now, when we talk about GI motility, the GI stands for gastrointestinal tract, or gastrointestinal tract motility. Now, when we talk about motility, we should remember that we are not talking about a region, but we are talking about a system, that is our gastrointestinal system. And these three motilities that is presented in your in this slide, that is called peristalsis, BER or basal electrical rhythm, and MMC or migratory motor complex. These three are the type of motility that is observable almost throughout the intestinal tract. Now, there are some regional motilities also, which is specific to organs, like receptive relaxation, antral systole. They are very specific to stomach. Then there are segmentation contraction and tonic contraction. They are specific to intestines. And there is mass action contraction, which is specific to your large intestine. So these are the type of specific or regional GI motility. So now we know that there are two type of GI motilities. One is the motility which encompasses the whole GI system, starting from the esophagus to the anus, and there are some regional GI motility which is specific to the system. Now, as my previous speaker, Dr. Barnali, has explained so nicely that how these how the tract is lined by two different layers of smooth muscle. So now we know that there are, so after her talk, we know that there are two type of muscle layer. One is the smooth muscle layer, which is towards the center of the GI tract, if I take a TS, and there is a longitudinal smooth muscle layer which is present outside the, or at the outer layer of the tract. Now these longitudinal smooth muscles are present through the long axis of the tube, and the circular muscles, they encompass the throughout the periphery, that we must remember. So now what happens here? So what kind of movement that we can see? Let's see in this diagram what we have. I tried to explain you that when the circular muscles or the circular fibers, they contract, they basically decrease the diameter of the tube. And when the longitudinal fibers, they shorten, so there are two things at the uh, along with the relaxation of the circular fibers also. So what happens? The diameter increase as well as the length shorten. So you can see that if there is a mechanism which can generate a contraction behind the bolus and relaxation in front of the bolus, or the chyme, or anything. So what will happen? That particular structure or the bolus will be pushed. It can move ahead. So this is the basic of your movement of gastrointestinal tract. So we'll start with peristalsis. Now the peristalsis is a Greek word. The origin is Greek. It came from the word peristalene. Peri means around, and if I say stellene, the stellene means uh, bring together or draw in. Now another dictionary is there which says that this word has come from the word peristaltikos, which means contracting around. So both the words, whatever they mean, maybe the meanings are a little bit different, but it actually says or depicts the essence of the peristalsis. So peristalsis is basically a movement which is made up of uh, alternate contraction and relaxation on the two side of the bolus. Now this movement is intrinsic. That means it is without any control of higher centers. It has a basal rhythm, and the speed of the peristalsis may range from 2 to 25 centimeters per second. Now, so basically what happens, as I said, it is independent of extrinsic innervation, and it's a kind of reflex. So what happens when the bolus passes through the tube, or anywhere, suppose in esophagus, or you can say in the small intestine. So what happens? That bolus or the chyme, they stretch the wall of the uh, tube. Now, when there is a stretch, the cells, they release one neurotransmitter called serotonin, which basically activates the myenteric plexus. Now we know there are two type of plexus in the enteric nervous system. One is myenteric plexus, another is Auerbach plexus. Now the myenteric plexus is associated with the contraction of the smooth muscles. Now what happens when this myenteric plexus is excited by the stretch? Now it behaves differently in the two zones. One zone, which is the retrograde fashion, another is the anterograde. Retrograde means behind the stretch. So in the behind the stretch, they stimulate the cholinergic neurons, and these activated neurons, they secrete acetylcholine as a primary neurotransmitters and as a co-transmitter substance P. Now these two, they basically cause the smooth muscle contraction, the acetylcholine. Whereas in an anterograde fashion, these cholinergic neurons, when they get activated, they activate neurons which secretes nitric oxide, vasoactive intestinal polypeptide, and ATP. Now, this nitric oxide and vasoactive intestinal polypeptide, they basically cause the smooth muscle relaxation. So now what happens here? So you get a push because of the contraction which is behind, and you get a relaxed state in the front of the foot. So when this waves, the peristalsis waves move from one end to another end, they keep pushing the bolus ahead. Now we must remember that this peristalsis movement is from the oral to aboral end. It is unidirectional. Now, this peristalsis depends upon a lot of factor. It is uh, basically the type of food that we are having, whether the food is wet or it is rough, it is dry. It depends upon that. It depends upon the size of the food. It depends upon the size of the bolus also. Sometimes we gulp a lot amount of food, and you'll feel that there is a severe constriction in your esophagus, and the food tries to go down, but it doesn't go. Now, in that case, you should remember whenever a peristaltic wave face a resistance that it cannot I mean, or it cannot go beyond, then the peristalsis wave stops. So, what are the factors that can modulate the peristalsis? The first thing is the mechanical characteristics of the gut content. As I said, the food, the chemical characteristics of the gut content, that means the food again, that what kind of food you are taking, the electrolytes, interference with ion channels, that means different drugs you can say, interference with different enteric hormones. These hormones, like few are few GI hormones are there, or gastrointestinal hormones are there, which can increase the motility, and there are some hormones which can decrease this peristaltic motility. and and obviously, though we say that it's an intrinsic mechanism of the GI tract, obviously there is an extrinsic control. That means the autonomic nervous system may control this peristaltic speed or the frequency. Now we know that these structures, they are made up of smooth muscles. So the both muscles, the longitudinal muscle and the circular muscle, both are made up of uh, smooth muscles. Now we should know some uh, salient features of smooth muscle contraction and smooth muscle properties when we are learning about the GI motility. The first thing we should remember that the resting membrane potential of smooth muscle is unstable. It is not stable like skeletal muscle. So it is unstable, and it varies from say in case of GI minus 65 to minus 45 millivolts, and so on. And just like just unlike basically the skeletal muscle, the spike duration, that is the action potential spike, it remains for a pretty long. In case of cardiac in case of skeletal muscle action potential, it is just like that. But here it stays for around 50 milliseconds when the spike is produced. As well as that these smooth muscle cells, they can show some pacemaker activity sometimes. Another thing that is very interesting here, since the smooth muscle uh, protein structures and skeletal muscle protein structures are different, and they are organized in different fashion. So what we find that the muscle contraction takes place a a little bit later. That means though the action potential is produced, to contract it, the muscle, you need calcium, and that calcium has to be released from the cellular organelles. So in between, and there the since the mechanism is different, since the carrier is different, and you need to activate the calmodulin for this calcium entrapment and all those things. So the spike and the contraction takes a little longer time than the skeletal muscle system. So it is around 200 milliseconds after the spike, whereas in case of skeletal muscle, after the spike, within 10 milliseconds the muscle contracts. And you will find that since in case of skeletal muscle they form cross bridge, and in case of smooth muscle they form latch bridge. So the piece and the peak contraction, it also takes a longer duration. So peak contraction takes place after 50 milliseconds. That means if you stimulate a smooth muscle, it will take a very long duration to show its peak contraction. So with this, as I said, that they may have some pacemaker activity. So we'll start with the first kind of another intrinsic rhythm, that is called BER, or basic electrical rhythm. Now this basic electrical rhythm, it starts from the stomach onwards. It is not available in esophagus. Now, as my previous speaker mentioned, that there are interstitial cells of Kajal that is present in stomach. They are basically the pacemaker of GI system. And as I mentioned earlier, that the fluctuation of membrane potential, so it fluctuates between minus 65 to 45 millivolt. So what we see, that continuously the membrane potential, it remains in minus 65, and then slowly it goes to minus 45, again it goes to minus 65, and it goes on like this. Now here we, as I mentioned, that BER is absent in two regions. One is the esophagus, as well as in the proximal part of the stomach, that is the cardiac part of the stomach, upper part. Now, in case of stomach and small intestine, we find these pacemaker cell at the outer border in circular muscles of the circular muscles, and the layer near the myenteric plexus, so that they can stimulate the smooth muscles via gap junctions. And in the colon, we find it at submucosal layer. Now, when we see this electrical activity or the BR, so what we find is that slowly as it reaches to the threshold level, then only there is contraction. I mean, there is formation of spike potentials, and again the membrane potential it goes down. Now, unless and until there is a formation of peak potential, you don't find any intestinal smooth muscle contraction. So the basically what we see, that the muscular tone, it slowly gradually increase, and then it falls again, slowly gradually increase, and again then it falls, and then ultimately when the action potentials are produced, the muscles start to contract. Again, it is very slowly. So what we find, that if I see a gross smooth muscle contraction, we find that the tone of the muscle increase over time. It takes a lot of time to increase, and then again it gets relaxed. So the whole system is that though the BR, or the rate of BR, and if I see the rate of muscle contraction, what we find is these two are different. It is not the same. So now we can appreciate that it is different in different part of the uh, GI tract. In case of stomach, the rate remains within at least 4 per minute. I mean, we get 4 contractions per minute. In duodenum, it is 12 per minute. In distal ileum, again, it reduces to 8 per minute. Cecum, it is increased in as 9 per minute, and in sigmoid, again, it is increased up to 16 per minute. Now, now again, this BER can be modulated by autonomic nervous system, where the epinephrine, it decreases the rate of BER, and if there is a parasympathetic stimulation and acetylcholine is released at the end of parasympathetic nervous system, then the rate of BER will be increased. So I think up to this, the things are very easy, smooth going. Now we are coming to another interesting movement of GIT. We call it as migrating motor complex. Now, when we say migrating motor complex, there are several names. The other name is migratory myoelectric complex, but actually it is the same. Now, what we see, the migration course is limited from stomach to distal ileum, and migration rate on an average, it happens around 5 centimeter per seconds. Now, if you ask me what is migrating motor, why we are saying it as migrating motor complex, then we should I should reply you that it's a cyclic recurring motility motility pattern that takes between two meals. That means you take one meal and the second meal. Suppose you have taken lunch and then you haven't taken anything, and ultimately you are going for dinner. So in between these two phase you find migratory motor complex. So this is not peristalsis, this is not BER, this is totally different, and basically this movement is required because they are the housekeeper of the intestine. Basically they clean up the intestine, and these complexes they generated at the interval of 90 minutes, and it is composed of three phases. The first phase, uh, if you can see the arrow there, the first phase is pretty long. It covers up around you say 40 to, here it is the first phase. So first phase, it is pretty long. It takes up almost the 40 to 60% of the total time. Then you get a second phase. In the second phase you will see some irregular activities, that there are some electrical discharges that can be observed. And again, this also covers around 20 to 30% of the total time. That is, as I said, it occurs in the interval of 90 minutes. Now after that, there is a regular activity. Now, whenever there is a regular activity or regular action potential is appearing in the smooth muscle, then the contraction takes place, and it is moving from one place to another place. Now, whenever there is a contraction, the circular muscles will contract, as well as the longitudinal muscles will contract. So anything which is left inside the intestine will be pushed to the next level. So in this regular activity, so these regular activity spikes, they basically they stay for around 5 to 10 minutes. Now I am not using the term percentage. Till now I was using the term percentage for stage 1. I am in the phase 1 and phase 2, but for phase 3 I am not using the term percentage. Rather, I'll be very specific that it is around 5 to 10 minutes. Now you can see that how these patterns or waves, they move from one place to another place, from stomach to the distal ileum. Now we are coming up to the regional GI motility, and we will be starting with the first, that is for the stomach, that is the receptive relaxation. Now you know that normally when the stomach is empty, the size is not very big, it is very small. But when you eat a lot of food, the stomach need to be relaxed so that it can accommodate the food. So receptive relaxation is nothing but it is the relaxation of fundus and the body of stomach. Now, so how it happens? So obviously, the higher centers, the vagal nuclei, the vagus, basically the parasympathetic activation is there, and these parasympathetic activities are basically triggered by the movement of pharynx and esophagus. So basically, when you are swallowing something, that stretches the muscles, that gives signal to your higher centers and the vagus, they start discharging those vagal fibers. They come to stomach and they relax um, the fundus, so that more and more food can be accommodated. So we call it as receptive relaxation. So this relaxation has taken place to receive the food. That is why it is called receptive relaxation. Now come to the antral systole. This is different. Now previously it was relaxation, now it is the contraction. Now it is the contraction of antrum, the lower part. Now, why the antrum need a contraction? Because the stomach has to mix the food with the acid, with the enzyme, so that the all your digestive things can be taken place. So now this antral systole is not mediated by the higher center. Rather, this one is mediated locally because of the peristaltic wave. Now, each time when the antrum contracts, it may last up to 10 seconds, and the wave occurs, and this contraction occurs 3 to 4 times per minute. So I think the diagram is visible, and you can appreciate whatever I have said till now, that how these mechanical stimulus in the pharynx, and then they stimulate the vagal center, and here it is the vagal center. So after the center sends signals to the stomach, and ultimately the size of the stomach, it basically increase. So I'm not going to the detail of these, because we are in a very general uh, topic. We are discussing a general topic, but there are different phase. When the food is there, the food is mixed. Now the food has to be sent to the next part, that is your small intestine, the duodenal part, and in between that there is a sphincter called pyloric sphincter. So, what happens when there's a contraction of the proximal antrum. That contraction, it forces the food towards the terminal um, deodorant, in particular this place, but the pyloric sphincter is still, it is closed, because if the food particle size are not less than 2 millimeter, it is not going to pass. So you have to grind it. You have to grind it to so fine that it can pass through this sphincters. Now the second part is the phase of emptying. So the phase of propulsion, it is because of proximal antrum. Now the phase of emptying, it is now the stomach needs to empty the food content, which is almost digested, till the stomach has the role. So in that case, it is the role of middle antrum. And then again, if the the pyloric sphincter is open, obviously because of contraction, the food may go, as well as there is a transpyloric movement, as well as there is a retrograde flow. That means the food may go to both sides. It can go in the stomach and churn back, and some amount of food that will go through the towards the duodenum. Now the phase of retropropulsion, in that case, again, the terminal antrum contracts, and because of this contraction of terminal antrum, now the jet-like backflow that takes place, and sometimes you may hear it also. You can hear if you want. So there are so three basically there are, because of this contraction, three things occur, or there are three phases in this whole antral systole. The one is the phase of propulsion, then the phase of emptying, and then the phase of retropulsion. So here, one reflex that remains, and it may be discussed later in detail, that is called gastrogastric reflex, where the stomach itself, it relaxes its own self to act as a better reservoir for the undigested food, and it gives time to the food to get digested. Now next come to the intestine. So after stomach, the two things we are into the intestine. That is, the first one is the segmentation contraction. Now you can appreciate this small animation that I have taken from internet, obviously. So what we see is that after, when the food is semi digested, the chyme comes to the intestine, and in the intestine you have to mix it with the different kind of enzymes, as well as you need to absorb whatever has been digested to the basic or primary molecules. So here what you have to that you need to increase the contact of the food with the mucosa. Then only the food can get absorbed, as well as then only the food will get better contact with the enzymes. Now, one thing we should see, that in this type of contraction, till now we said that peristalsis movement, force the food from oral end to aboral end. But here the movement is retarded. As well as during the segmentation contraction, the food may go to and fro. That means it can go back, but not, it will not go back to the stomach, but within the intestinal segment, it may move backward as well as forward. And because of that, more mucosal contact, more activity of the digestive enzymes, and a better absorption. Now, the next type of movement is called as tonic contraction. Now, in case of tonic contraction, so what we find is, because of tonic contraction, the circular muscles, they contract so much that they basically separate the intestines into different chambers, and they remain it for longer duration. So basically, it isolates one segment of intestine from the other, and it is for a longer duration. As I said, that the smooth muscles, they take a lot of time to contract, as well as when they are contracted, they take a lot of time to relax also. So their movement is tonic, not phasic, particularly during this time. Now you can ask that how can we, just like heart, we do ECG or electrocardiography. Now, these movements and movement related disorders can be identified by another technique, that is called electrogastrography, which is again, so in this electrogastrography, we use this technique to measure these muscular movements or the electrical activities by placing different electrodes throughout the region of the stomach, and then the doctors or the gastroenterologists take the decision if there is any problem or not. Now come to the large intestine uh, part. So we know that this is called mass action contraction. So it is very specific to colon, and it helps. So in mass action contraction, the whole of the colon, they contract together, so the all the content of the colon, it is pushed to the rectum. Now these things, or this mass action contraction, it is not, it takes around 3 times. It happens 3 times per day, that the all content of the colon is pushed to the rectum, and one may feel for defecation after food, because the intestine prepares itself for the next set of food that is coming. Now, as I said that, and my previous speaker also mentioned that intestine has got its own brain, otherwise called belly brain or enteric nervous system. So they have their own intestinal reflexes. Now, these reflexes are of 3 types. Now, the 3 types are the reflex that are integrated entirely within the gut wall of enteric nervous system. That means it is not going to spinal cord, it is not going to brain, it is whatever is happening within the spinal cord only. Just like peristalsis, the whole movement, you stretch the muscles contract or relax, it is within the gut only. Now, another thing is the reflexes from the gut to the prevertebral sympathetic ganglia, and then it comes back to gastrointestinal tract. So this one is another reflex, another type of reflex. And the third type of reflex is the reflexes from the gut to the spinal cord or brainstem, and then back to the gastrointestinal tract. That means what we find here, that these reflexes which are associated with the gastrointestinal tract. Now you know what is reflex. Reflex is an involuntary response or involuntary spontaneous response to any stimulus. So these reflexes can be of 3 level. That one within the organ, up to the spinal cord. So before spinal cord, it ends. It ends within the prevertebral ganglia. That means it is not going into the vertebra, but the whole circuit it completes before the going into the spinal cord. And the third one is going to the either spinal cord or vagal. So whenever I said that these are vagally mediated reflexes, this is happening because of vagus stimulation. So this is the third type of reflex that is happening. Now, when we count about the reflexes of GI system, there are 3 very common reflexes. One is enterogastric reflex, gastroileal reflex, and gastrocolic reflex. Now, let us decide first with the enterogastric reflex. Now, in case of enterogastric reflex, what we see is that this reflex is totally mediated by the two things. One, the content of the food, and the volume of the food. So presence of fatty acid, or the fluid is very high, I mean the chyme is hypertonic, or since the chyme is just entered the you say duodenum from the stomach. So in the stomach we know that there are a lot of HCl, so pH is very low, so it is acidic, the chyme is acidic in nature. So they will stimulate the receptors which are present on the duodenal mucosa. Now, these receptors, they basically are the duodenal enteroendocrine cells, which can release different kind of GI hormones. Then there are chemoreceptors and stretch receptors. So, stretch receptors, they identify the stretch. Now, the chemoreceptors, they basically identify the pH of the content. So, they use two different pathway. One is the long reflex, which go to CNS. It increases sympathetic system activity, it decreases the parasympathetic activity. And by the short reflex, that is within the gut only, the first type of reflex, the short reflex, which stimulate the enteric neurons, and they basically decrease the contractile force of the stomach. And so when the contractile force of the stomach is decreased, then less amount of food will, or the chyme will come to the duodenum. So duodenum will get enough time to absorb, I mean, the to neutralize the acid, as well as to absorb the fatty acid which is present. So this is the enterogastric reflex. Now, in case of uh, gastroileal reflex, now when there is a food in the stomach, so basically it initiates the peristalsis in ileum. Now when the so when there is peristalsis in ileum, that leads to the opening of ileocecal valve, and the food, it leaves the ileum, and it goes to the semi digested, or uh, almost fully digested food, food, no, so chyme. So it moves, so it is almost now near to be processed or called as feces. So they send it to the large intestine for a rest of the processing. So this is called gastroileal reflex. So when you are having food, the relaxation of ileocecal valve, the from the content which is getting digested in the ileum, it has been shifted, so the ileum and is free to receive the second batch of food. Then the gastrocolic reflex. Now, so by the name it suggests, it has a stomach component, it has a colonic component, and it's a reflex. So same thing, what happens, as I said, the mass action contraction. So that, so basically what we find, so whenever we eat something, whenever the food goes inside the stomach, the rest of the intestine, they prepare itself to receive the food. So they basically pass their content to the next order. So in case of gastrocolic reflex, so what you find is that as soon as you have food, that food, or the stomach release the food from the stomach to the duodenum, there is a mass action movement in ascending colon, descending transverse colon, and the descending colon, and the content is shifted to rectum. Now you may have observed that particularly in children, they defecate during the feeding time. So that is basically the example of gastrocolic reflex. So so with all these, starting from the very basic and to the reflexes, um, I think I'm at the end of my topic, and I may be a little bit fast. It may appear that I'm a little bit faster, but since in a single lecture we have to compile everything, so not everything I have told in detail. So anyone, if is having any questions, you can directly mail to me. My email address is Arjun Maitra, ARJUNMAITRA @gmail.com, so that I can answer you. So at the end, I'm again very thankful to my teachers, my Dr. Prasun Naik, that he has given me the chance to come in front of you and discuss a little bit of physiology that I also have learned from him only. So I always say, because he was my postgraduate teacher, so he built me. I think I have done some amount of justice to his hard work, because I was a very hard nut to crack basically every time. So thank you everyone. Thank you very much for being with me, for listening to this small piece. Uh, Good night.
Thank you, Dr. Arjun. It was so nice. We have touched almost every part. So it's a nice presentation. And our target was to overview, give an overview related to the structure, to the functions, and obviously with a special emphasis to the gastrointestinal motility. And I think we did justice to that. With both of you have done splendid job. We have some feedback also for you. Sanchari Chakraborty has mentioned that you have done wonderful talk, and then we received some others also. Sarita Kanoria to thank you. Uh, Thank you, uh, Dr. Arjun, and thank you for mentioning. Uh, yeah, it's nice that both these. Dr. Barnali, I am putting you on stage also. Yeah, both
Chotu,
Dr. Barnali and Dr. Arjun, both of them, they started their PG with me. It's really nice to see both of you in this platform. And thank you on behalf of Physiological Society of India, the Education Committee of Physiological Society of India. Thank you very much. And if we receive any feedback regarding any comment or any query, particularly comments, we are getting good, good, in fact, uh, good support, good feedback we are getting from the persons. So, in case we get any query from to our side, we will let you know, and we will expect that you in the comment section you answer for them. Thank you very much for your time, for your support to the education committee and continuation with this uh, lecture series. Thank you. So, uh, anything you would like to share, both of you, Dr. Arjun and Dr. Barnali?
No, sir, it is over now. Thank you.
Dr. Barnali? Okay. Thank you. Thank you very much. So, we had a nice session on gastrointestinal motility as well as the overview of the digestive system. In this regard, what we have discussed, we heard that a name, Dr. Cajal, Cajal, a Spanish neuroscientist who was also known for his histology and neuroanatomical activity research. Dr. Ramon Y. Cajal, he received his Nobel lecture. He actually shared his Nobel lecture in 1906 with Camillo Golgi. So these two neuroscientists Golgi and Kahal, they are throughout our physiologic system. We learn about their work, and they have done a great job to the physiology, to the medical science. Next session on the lecture series we will continue, and soon we will announce that we have Dr. Kausik Bharati on board. Let us see. Dr. Kausik, welcome. If you would like to share something, please unmute yourself.
Yeah, excellent talks, both of them. And I'm very happy to, I learned a lot of things from there also, and lot of revision also. I got from this, highly refreshing. And I thank both of the speakers, Dr. Barnali and Dr. Arjun, both. Thank you.
Thank you, Dr. Kausik. Uh, so we'll be waiting for our next series of next session of this PSI online series. We would like to have our audience to have their feedback in this, and we have shared our registration link also. We can have the PSI membership also. They can communicate about their requirement, what type of lectures, what type of sessions they would like to have. That will be a help us to decide our next sessions. So that's all for today. We had a very good session on digestive system. Hope you have enjoyed. Thank you. We express our sincere thanks to Physiological Society of India, the members of the Education Committee of PSI, and also the technical support from Dr. Sompa and Dr. Vinay. Thank you very much. Thank you. Thanks a lot to everyone. Have a good night. Thank you. Stay tuned for our next series, which will be in 2024, January 4th Saturday. Thank you, everyone. Good night.